Welcome to our second edition of Short Talks. Today we have four fantastic speakers who I will introduce right now. Our first speaker is Hemant Sistla, who completed his Master's in Physics at the Sir Sathya Sai Institute of Higher Learning in Puttaparthi, India. He is currently a scientific associate at thinkMolecular Technologies, a molecular simulations company that is based in Bengaluru. Our second speaker today is Cyrille Kounde, who completed his BSc degree in biochemistry at Paris Diderot University and his master's in medicinal chemistry at Paris Descartes University. He started his career in industry as a drug discovery scientist at Evotec and then joined Novartis UK in 2010, later moving to Novartis in Singapore. He is now pursuing a PhD at the Imperial College in London. Our third speaker today is Luke Simpson, who graduated his medical sciences degree at the University of Aberdeen. He then joined Professor Gopal Sapkota's lab at the MRC at the University of Dundee. And Luke very recently successfully defended his PhD. So congratulations to Luke. And our final speaker today is Alexander Hanzl, who obtained his BSc in chemistry and then moved into chemical biology for his master's at the FMI in Basel. Alex is now a PhD student in Georg Winter's lab at the Center for Molecular Medicine in Vienna. So now let's hear from the trainees.
Greetings to all who have joined with us today. I'm Hemant Sistla, working as a scientific associate at thinkMolecular Technologies Private Limited, Bengaluru, India. thinkMolecular is a molecular simulations company with its focus in drug discovery. At the very outset, I would like to thank our founder, Dr. Hemant Sistla, for his invaluable inputs in this work. Today, I'll be talking to you all about the RNF114-BRD4 PROTAC ternary complex modeling. In 2019, Spraden et al. Identified a naturally occurring compound, nimbolide, as a ligand of the ligase RNF114. In this study, they demonstrated the degradation of the bromodomain protein BRD4 using nimbolide, which was incorporated in a PROTAC. Now in 2021, Luo et al. From the same group identified an analog of nimbolide, EN219, using chemoproteomic approach. And this was also deployed in a PROTAC to target the same protein. This is the structure of the PROTAC developed by Luo et al. This is the EN219, which binds to the E3 ligase. And this is the warhead of the PROTAC, which binds to the target. Now, for the rest of my presentation, I'll be referring to this PROTAC as compound 1. Now, the objective of my work was to create a ternary complex model of RNF114, BRD4, and compound 1 using protein-ligand docking, protein-protein docking, and molecular dynamics simulations. Now, RNF114 is a 228 amino acid E3 ubiquitin ligase with a mass of about 26 kilodalton. Now, the thing is that a 3D experimental structure of RNF114 is not available. Now, this here is the image of the blast output of RNF114. So this bar here indicates the sequence identity of these various proteins with that of RNF114. Now, as you can see, the sequence identity of many of these proteins with RNF114 is quite decent, but then the sequence overlap is poor, and therefore these proteins cannot be used for building a homology model of RNF114. Now, even the best among these results, RNF125, whose domain diagram is shown here with RNF114, even this cannot be used for building an homology model for RNF114. Because the available structure of RNF125 does not have the entire sequence overlap with the, does not provide the entire sequence coverage with respect to the RNF114. Now, since an experimental structure nor a homology model of RNF114 was accessible, we chose to utilize the structure reported in AlphaFold. So RNF114 is a zinc binding ligase. That is, it has 5 zinc fingers, of which 2 are present in the ring domain itself. Now, zinc atoms were incorporated into this AlphaFold structure by taking the structure of RNF125 as the template. The figure on the left shows the alpha fold structure. Now this region here, the N-terminal region marked in orange, indicates that the confidence of the model for this particular region is quite poor. Now, according to the literature, this is the cysteine where the EN219 and the nimbolide get covalently attached. But this region does not form a pocket where the ligand can make meaningful interactions. And therefore, we had to subject our RNF114 to 100 nanosecond molecular dynamic simulation so that the terminal regions, the N-terminal and the C-terminal regions, can fold and make the structure compact so that the ligand can have meaningful interactions. Now, here on the right, you can see the trajectory of the 100 nanosecond molecular dynamic simulation that was run using GROMACS. This trajectory was then clustered, and the center of the largest cluster was taken forward. So this is the chosen structure of RNF114. As you can see here, this is the pocket where EN219 is supposed to bind, and this is a cysteine 8 where it gets covalently attached. Now, RNF114 was used as the receptor onto which EN219 was covalently docked. Now, this was done using the software suite ICM, which is provided by MolSoft. So this figure here shows the key interactions made by the EN219 on our protein. Now, the regions in green represent the hydrophobic regions, while those in gray are the ones that are exposed to the solvent. So as you can see, this part of the EN219, the bromophenyl, is buried into the hydrophobic pocket, and the other bromophenyl is exposed into the solvent. Now, this is where the linker is attached for a PROTAC to be constructed. From our studies, we also concluded that the R isomer of EN219 is likely to be active on RNF114. Now, allow me to refresh your memory of our compound 1. So this is the EN219, which binds to the E3 ligase RNF114. And this is the warhead JQ1, which binds to the bromodomain protein BRD4. So please bear with me as I explain to you the significance of this crowded picture. So to generate the binary complex, the X-ray structure of JQ1 bound to BRD4 was docked onto the arrived structure of RNF114. So this is the structure of the RNF114. It was treated as a receptor onto which the BRD4, the BET1 domain of BRD4 was docked. So this was treated as a ligand. So during this process, about 10,000 binding modes were generated. Now this was done using the protein-protein docking module of ICM. So as 10,000 modules, different binding modes were generated, some of those modes are represented here. So these 10,000 binding modes were then clustered basing on the interaction fingerprints. So these interaction fingerprints are nothing but a representative of the different patterns in the interaction between the bromodomain protein and the RNF114. So the regions here, you can see the different colors here. All of these represent the different clusters. So what we did was we took clusters from these clusters, and then basing on the docking score, the interaction energy, we shortlisted these cluster centers. And then we got a couple of binary complexes. Now, these were again shortlisted based on the protactability of these individual ligands. So these ligands here, so what I mean by protactability is that basing on how close these ligands are, what are their orientations, whether they can make the PROTAC which we have designed. So basing on these considerations, we have shortlisted them further to about 50 binary complexes. Now, these potential binary complexes were then further subjected to energy minimization and short ND runs using OpenMM. So the binary complexes arrived thus were then subjected to this PROTAC docking. So that is our compound 1 was covalently docked onto this binary complex RNF114 and BRD4. So the docking modes of the PROTAC were assessed based on the key interactions that the individual ligands EN219 and JQ1 made with the respective proteins. So as you can see in this picture, this is the compound 1, and this is the EN219 part of the PROTAC, and this is the JQ1 part of the PROTAC. So this binds to our E3 ligase RNF114, and this is the part which binds to the PET1 domain of BRD4. And this is the linker which joins the two ligands and completes our PROTAC. The figure on the right indicates the interaction diagram. That is, it indicates the key interactions which are PROTAC, which are compound 1, makes with the binary complex. Now, the regions here, these regions in the green, as I mentioned earlier, they indicate the hydrophobic regions, while those in the gray are the ones which are exposed to the solvent. Now, to summarize what the work which I have done, so this workflow succinctly summarizes the efforts taken up during this work. Now, in which a functional version of RNF114 was built. Then the active chiral conformation of EN219 was identified, which was then docked onto the RNF114 structure. Then a binary complex of RNF114 BRD4 was obtained using protein-protein docking. Subsequently, the compound 1 was docked onto this binary complex to make the ternary complex, which was the objective of our work. Now, in this process, protein-ligand docking, protein-protein docking, molecular dynamics, and trajectory analysis were the key techniques and tools that were employed. So we look forward to putting the ternary complex in perspective by introducing ubiquitin-bound E2 ligase into the picture. So this way, we hope to gain structural understanding of the feasibility of ubiquitination of the target. We are also very keen on using our workflow on building more ternary complex models and gain useful structural understanding for the prospective designs of the PROTACs. Now, this kind of work is being keenly pursued at thinkMolecular. So as you can see here, this is the androgen receptor, and this is the E3 ligase cereblon. And this PROTAC here is ARV110. So ARV110 is a clinical candidate which is currently in phase 2 clinical trials, and this has been designed by Arvinas, and this is an androgen receptor degrader. So this is a ternary complex which we have built, which we have modeled, and we are quite sure of its. We are quite confident in its structure, and we do hope to compare this with the experimental structure of this ternary complex whenever it is available. Now, I thank Dana-Farber Cancer Institute for giving me this opportunity, and I thank each one of you for your very patient listening. Now, you can write to us at info@thinkmolecular.in for the coordinates or any other queries that you might have. Thank you again for your patient listening.
Good afternoon, everyone. My name is Cyrille Kounde, and today I would like to talk to you about the conditional control of PROTACs, which are called the art of taming the chimeras. So as an introduction, let me start with this. So we recognize that the PROTACs are now efficient tool to knock down the protein, but also we are seeing that in academia and industry as have been used as a therapeutic or new therapeutic modality. So if we consider PROTAC as a therapeutic modality, we know that they can be highly specific for the target, but at the same time, there is a possibility of having both on and off target effects from PROTACs due to the duration of action, but also to the site of action where the PROTAC is eliciting its activity. So considering those potential side effects, the question we had was, can we control when and where to knock down a protein with PROTACs? And the idea or the remit of this question was already, can we develop chemical tools that will allow protein degradation to be done with added level of precision. So to answer this question, we came up with two approaches. The first one was to use light as a precision tool. Light has some properties that are very useful, such as it can be delivered very fast, it is non-invasive, and there are a few features that you can control, such as the area, the duration, and the intensity of the light. So the second approach we decided to use was to look at antibody PROTAC conjugate. So can we conjugate a PROTAC to an antibody in order to have specific tissue delivery of a PROTAC? So this is what I'm going to cover in the next 5 to 10 minutes here. So first, the first approach was to look really at cage PROTACs. So the idea was to attach onto the PROTAC a caging group, which is a photoactivatable. And this caging group actually will render degrader inactive. Now, upon light irradiation, this caging group can be removed and then will release the active degrader, which can then form the ternary complex and trigger the degradation of the protein. Now, the good thing about a system like this is that you don't need to engineer the UPS system itself. You don't need to modify the protein, but only have a small chemical modification of the degrader itself. Now, our prototype degrader was a BRD4 degrader. What we did is that we used the hydroxyproline from the VHL ligand, and then we block or we cage this hydroxyproline with the DMNB group, dimethoxynitrobenzyl, that prevented the interaction with a key amino acid. So this cage molecule in itself is inactive or should be inactive. Before testing the molecule in cells, we looked at first at the photochemical properties, and Dr. Charlie Sanders developed a small UV box for us that allows us to uncage this PROTAC3 and to check by LCMS efficient uncaging. So as you can see, after 3 minutes, we have complete uncaging of the PROTAC, and this was nicely done in vitro. So next, we decided to move into cells. So here on the left, what you can see is a parent degrader, which is fairly active. As you can see, can degrade BRD4 very nicely. So what about the cage degrader? So in absence of light, those are the blue rectangle here. You can see that this PROTAC is completely inactive, regardless of the concentration. But as we shine light on this on the cells, only for 1 minute, you can see nice dose-dependent degradation of BRD4. We also looked at cell proliferation. So how does the PROTAC affect cell proliferation before and after irradiation? So if you look at the parent degrader, so PROTAC2, we have a nice cytotoxic effect whereby the cell actually die upon being treated with PROTAC2. Now for PROTAC3 before irradiation, we have what we call a cytostatic effect, where the cell just stop growing, while after irradiation, we have a nice overlay with PROTAC2 itself, meaning that we have effectively uncaged the PROTAC to achieve cell death in this case. Dr. Masha Shepinova also developed for us a BRD4 GFP construct that we overexpress in EC293 cell line, and we'll be able to show that upon irradiation, again, we can have nice degradation of the GFP BRD4 construct. So we're not the only one to work on light activated PROTACs. So a few papers have been published not only on photocage PROTACs, as I've just shown, but also on photoswitchable PROTAC, which is a more reversible type of a photoswitchable PROTAC. So we believe that there is an opportunity here using light activated PROTACs, not only in chemical biology, but also if the properties of those PROTACs are being worked out nicely, there might be an opportunity also in photopharmacology. The second thing I want to talk about are targeted delivery of PROTACs with antibody. So you can imagine having two cell lines whereby one cell line poorly express a receptor of interest, but has a good expression of the protein of interest. And another case whereby the cell has a very good expression of the receptor of interest and also a good expression of the protein of interest. So you can imagine that we can potentially use this different level of expression of the receptor to target the cells with an antibody. And the idea was really to conjugate a PROTAC to an antibody here. And the expected outcome would be that in one case, we'll have internalization or after recognition of the antibody, of the receptor, we'll have internalization and release of the PROTAC into the cell, where on the other case, this will not happen because the cell doesn't express the receptor on the surface. So this work has been done with Dr. Maria Monero and Nafsika Forte, whereby they took trastuzumab, reduced the disulfide bond, and then conjugate the free sials with this dibromomaleimide, which is a nice tool developed by James Baker's lab. And then Maria also made this azido PROTAC here, and it was then just a question of coupling this azide with this alkyne that we have on the BCN moiety via spring-promoted azido-alkyne cyclization. So once this was done, we had a conjugate in hand. As you can see, as the cleavage site was here an ester. So we also wanted to make sure that the conjugate in itself has sufficient stability on its own before treating the cell. So this is what we've done. So incubating this conjugate in PBS at 37 degrees over the first 6 hours, we observe very minimal degradation or minimal hydrolysis, of this conjugate. It's only after 24 hours that we had a bit more hydrolysis of the conjugate. Nonetheless, that was good enough for us to move forward and test the compound in cells. So we've used 4 different cell lines. So 2 HER2 negative and 2 HER2 positive cell lines that you can see here from the gel. The receptor status was also confirmed by immunofluorescence. And we also made sure that the 4 cell lines did express BRD4. Initially, we first tested the parent PROTAC in those 2 cell lines, or in those 4 cell lines. So as you can see, the parent PROTAC does degrade BRD4, both in the HER negative and HER2 positive, although the HER2 positive seems to be more sensitive here. As you can see that there is more degradation on that side. We also did a washout experiment whereby we treated the cell only for 1 hour, and then we washed out the conjugate, so the PROTAC itself. And as you can see here, we have much less degradation after treating only for 1 hour versus the 4 hour on the upper panel. So the key results came here when we treated the cell with a conjugate for 4 hours. So in the HER2 negative, we didn't observe any degradation of BRD4, while in the HER2 positive cell lines, we did observe nice degradation. So that was a key result here that showed great selectivity and that the approach was feasible. We also did the same washout experiment as previously, whereby we treated the cell for only 1 hour and washed out the conjugate. As you can see in the HER2 negative, we didn't observe any degradation, where we did have degradation in the HER2 positive, although it was less pronounced than when we treated for 4 hours. So altogether, this antibody PROTAC conjugate has also been pursued by notably Genentech, where they've shown that those conjugates can be used in vivo to improve PK parameters. So we believe that there is a huge potential here to really develop novel and selective targeted therapy by conjugating the PROTACs and antibodies. With this, I would like to thank the people that were involved in the work. So at Imperial College, Professor Ed Tate, Maria Charlie, and Maria Monero, who have been involved in the cage PROTAC work and also the antibody work. Professor Jamie Baker, who developed the conjugation strategy for the antibody. And finally, the sponsors, GSK and EPSRC, who funded those research. With this, I would like to thank you for your attention, and I would be happy to take any questions.
Hello, I would like to thank the organisers for the opportunity to present at today's trainee short talk session. My name is Luke Simpson, and I recently finished my PhD project with Professor Gopal Sapkota at the Medical Research Council Protein Phosphorylation and Ubiquitination Unit at the University of Dundee in Scotland, where my project work has involved the development and application of a ligand-inducible affinity-directed protein missile system for targeted proteolysis. So first, if we consider proteolysis targeting chimeras, referred to as PROTACs, which have been successfully developed for the targeting of various proteins for proteasomal degradation. And typically, PROTACs are heterobifunctional molecules that consist of two distinct chemical moieties. For example, one that targets the protein of interest, abbreviated to PY for this talk, and the other that recruits an E3 ubiquitin ligase and connected by an optimized linker. That in cells, these molecules would bind to the PY with one moiety and bind to the E3 with the other to form an active PY PROTAC E3 ternary complex to bring the E3 into close proximity of the PY, mediating PY ubiquitination and subsequent proteasomal degradation. However, currently, robust ligands for a substantial percentage of the human proteome remain to be developed. Therefore, various tag-based PROTAC technologies have been developed in order to determine the degradability of a target protein, but then, and also to determine the potential benefit of degrading that protein. For example, through the evaluation of the biological phenotype following the removal of that target protein, and also for the study of protein-specific biology. And for example, the labs of Alessio Chile and Dario Alessi here in Dundee have designed and characterized various halo tag recruiting PROTACs, including their lead compound halo PROTAC E, which consists of a halo tag recruiter and a VHL recruiter for the targeting of PUIs that have been tagged with halo for proteasomal degradation. However, the knocking in of a non-fluorescent halo tag homozygously onto a PUI using CRISPR-Cas9 gene editing technology can prove challenging. Therefore, to overcome these challenges, we aim to develop a system that exploits protein-specific small polypeptide binders, such as nanobodies or monobodies, that following their expression in cells, specifically bind to a PUI with high affinity, and that we could we aim to conjugate these binders to the halo tag with a glycine linker. And we term this approach the ligand-inducible affinity-directed protein missile system, or the L-AdPROM system for short. And for initial system characterization, we first looked at targeting proteins, target proteins that had been tagged with GFP. And in order to do so, we designed a halo-AdPROM construct consisting of the halo tag conjugated to a previously reported anti-GFP nanobody and tagged with a flag reporter for detection. That following the expression of this halo-AdPROM construct in cells by retroviral transduction would bind to the GFP tagged POI with high affinity, and that treating these cells with halo PROTAC E would bind to the halo tag component of the AdPROM system, while simultaneously binding to VHL, mediating the recruitment of the Kounde CRL machinery and promoting the POI ubiquitination by proximity and leading to POI proteasomal degradation. And so first we looked at targeting ALK1 for degradation through the AdPROM system, halo-AdPROM system. Now, ALK1 is a serine-threonine protein kinase that is involved in the regulation of the initiation of autophagy, and to our knowledge had not been previously targeted for degradation using PROTAC-based technologies. So following the expression of the halo-AdPROM system in ALK1 homozygous GFP knock-in cells, we treated these cells with increasing concentrations of halo PROTAC E for 24 hours and observed a concentration-dependent reduction in both GFP ALK1 levels and flag-tagged halo-AdPROM levels at nanomolar concentrations. We then treated these cells with 250 nanomolar of halo PROTAC E and observed a time-dependent reduction in both GFP ALK1 and flag-tagged halo-AdPROM levels over 24 hours. So following the targeting of various GFP tagged POIs for degradation using the halo-AdPROM system, we next then wanted to determine the versatility and applicability of the system. So we substituted the anti-GFP nanobody component of the halo-AdPROM system for a previously reported A-HRAS monobody that binds oncogenic H and KRAS isoforms, and that in the presence of halo PROTAC E would mediate the ubiquitination and proteasomal degradation of untagged endogenous RAS proteins. So following the expression of the flag-tagged halo A-HRAS AdPROM system in A549 lung adenocarcinoma cells, we observed a concentration-dependent reduction in both pan-RAS and flag halo-AdPROM levels after 24 hours at nanomolar concentrations of halo PROTAC E. And in various RAS-driven cancers, mutated hyperactivated RAS promotes constitutive activation of downstream MAP kinase and PI3K signaling to promote cancer cell progression. So following halo-AdPROM mediated RAS degradation in these cells, we then wanted to investigate downstream RAS-driven signaling. And we observed a reduction in EGFR phosphorylation at tyrosine 1068 and a time-dependent reduction in AKT phosphorylation at phosphoserine 473. Therefore, this data suggests that following halo-AdPROM mediated RAS degradation, we can inhibit RAS-driven signaling. So to summarize, we've developed a halo-based ligand-inducible AdPROM system for the targeting of proteins for proteasomal degradation. And the halo-AdPROM construct consists of a protein-specific binder conjugated to the halo tag that, following expression by cells, for example, by retroviral transduction, binds to the target protein with high affinity. And that treating these cells with halo PROTAC E, which consists of a halo tag recruiter and a VHL recruiter, promotes the ubiquitination of the target protein, leading to the target protein proteasomal degradation. And through substitution of the target protein binder, the halo-AdPROM system is versatile and adaptable and can be employed to study the biological function of a target protein. And with that, I would like to thank my supervisor Gopal and the rest of the Subquota group for all of the help and support over the course of my PhD project. and the Chile group and the Ghana group for the help with the L-AdPROM project. And also I'd like to thank the services provided by the MRC PPU and the Dundee University School of Life Sciences for making this project possible.
Hello and welcome. My name is Alex. I'm a PhD student in the Winter Lab at the Center for Molecular Medicine in Vienna. And first of all, I'd like to say a huge thank you to the organizers for giving me a chance to present some of my PhD work at this seminar series. Now, for this project, we actually started out being interested in resistance mechanisms. And of course, in conventional inhibitory pharmacology, several resistance mechanisms have already been identified, and many of those would translate to targeted protein degradation. However, the fact that targeted protein degradation is dependent on multi-component protein assemblies and cascades throughout the cell means that there's likely also novel rules and routes for resistance. And we actually started out taking a shot at this by comparing two different E3 ligases. On the one hand, we looked at the Cal4 cereblon E3 ligase, and on the other hand, to the Cal2 based VHL E3 ligase. And for both of these E3 ligases, we had a set of compounds for cereblon DBET6 and for VHL R771, which were highly similar in their potency and degraded the same PET bromodomain family proteins. Now, the reason why these two E3 ligases were especially interesting to us is because they're highly divergent in their essentiality profile. While most cell lines don't really care much about losing VHL, the same doesn't hold true for VHL, which is a common essential gene throughout most cell lines. Now, we first looked at this problem by comparing resistance rates and probabilities for these two different compounds that I mentioned here. And we could identify that for the cereblon E3 ligase that is non-essential, we had about a 10-fold increase in probability of resistance and the rate of resistance. We attributed this to potential genetic defects that arise in these resistant clones. And to measure this or to look at this more closely, we utilized a method called hybrid capture, where we generated resistant pools of cells and in these pools enriched for target genes, a set of 25 different target genes. After enrichment, we could sequence these genes for potential mutations or genetic alterations. And when we did this for these two different degrader compounds, we actually found that for cereblon, most of the position mutations and alterations happened within the substrate receptor cereblon itself. While for VHL, we got a much more split image where half of the positions, half of the mutations were actually in the substrate receptor itself, while the other half was actually in the Calin backbone. Now, at this point, we looked more closely where these mutations actually lie, and we found that a large portion of these mutations actually lie very close at the interface of the ternary complex that is generated. And for us, this really sparked an interest in how mutation of the E3 ligase could actually manipulate and affect the ternary complex formation and therefore the potency of degraders. And to take a more systematic approach at this, we utilized the method called deep mutational scanning, where we selected positions on the VHL and cereblon substrate receptor that are in close proximity to the degrader binding site. And we mutated each of these positions to every other possible amino acid. This generates libraries of around 1,500 variants, which can then be overexpressed in knockout cells of these substrate receptors. And through chemoresistant screening, we could selectively enrich for mutations that have an advantage over others and identify this via sequencing. And we took a first approach at this by comparing several different VHL-based degraders, 4 of them being the PET bromodomain degraders, and one of them being a SMARCA2/4 degrader. And when we looked at the mean of and the average over all of these different degraders and positions, we could localize actually the consensus resistance closely to the binding pocket of the VHL ligand, which makes a lot of sense because all of these degraders that I mentioned here actually depend on the binding of this VHL ligand. However, of course, the power of this method lies within comparing two different degraders and looking at differences that might be present there. And to take a shot at this, we compared the SMARCA2/4 degrader versus the BET degraders. And we could identify a set of positions or hotspots that as we call them, that would be uniquely required for SMARCA2/4 degradation while being indispensable for BET bromodomain degradation. And we next wanted to validate these, of course, and I'm just showing you here two positions and two mutations, where in both cases mutation leads to actual resistance to ACBI1, so the SMARCA2/4 degrader, while not affecting the BET degrader. Interestingly, for this histidine 110 position, when it's mutated to a leucine, we actually identified a sensitivity shift in the BET degrader, R771. So this was very interesting for us. And we took the next step by looking at target degradation. And we could identify that for the ACBI1-based degrader, we could nicely recapture the resistance also on target degradation level of SMARCA2 in this case, while BRD4 degradation was not really affected. At the same time, for the position histidine 110, we could see that we could again capture the resistance on SMARCA degradation, on top of which we could also identify the sensitivity to BRD4 degradation based on target degradation, BRD4 degradation. Now, at this point, we teamed up with Alessio Giuli's lab, and a PhD, Ryan Casement, in his lab actually performed biochemical validation via fluorescence polarization, and could also identify or attribute this resistance shift in this asparagine 67 mutant to loss of ternary complex cooperativity with SMARCA4. Now, at this point, I want to switch to our cerebrum-based degraders, where we actually did a comparison of a molecular glue type degrader called CC9009, which degrades GSPT1, and the DBET6 BRD4 PROTAC that I've already mentioned earlier. Again, we could identify several positions that had selective, several hotspots that had selective resistance to one or the other. And I'm just showing you here a validation for one of the molecular glue specific positions. And nicely, again, when we generated this single point method, we could nicely validate this resistance to the molecular glue degrader while not affecting the BET PROTAC. This again translated to loss of target degradation, which we could nicely see on GSPT1 levels. And at this point, we were actually very interested and very happy to see that we also captured a position that was actually identified in pomalidomide refractory multiple myeloma patients via sequencing. So this histidine 57 position was very recently identified. And when we generated this point mutant, we actually could see rescue of degrader potency back to knockout levels of the substrate receptor. And the same held true for target degradation for both GSPT1 as well as the BET bromodomains. Now, with this, I hope I could convince you that essentiality of the E3 ligase shapes the rate and the pattern of resistance mutations, and furthermore that our deep mutational scanning method can identify substrate and degrader specific mutations that can lead to identification of novel resistance mechanisms, as well as to potential degrader improvement and optimization. Now, I want to say here that we are actually also close to submitting these libraries to Etchin, so I hope that anyone in the community will soon have the chance, should they want to utilize this method. With this, I'd like to thank the team. So from the Winter Lab, Sophie, Matthias, Ele, and Hanne have been highly influential on this project. Of course, Alessio and Ryan have been also very helpful. And with this, I'm thankful for your attention, and I'm happy to take any questions, and I'm already looking forward. Thank you.